Hi, my name is Aleksandra. Unit 6 talks about plants. Chapter 36 focuses primarily on the behavior of flowering plants. Section 2 of Chapter 36 is on plant hormones. Plant hormones. There are several major types of plant hormones. These include auxins, cytokinins, gibberellins, ethylene, abscisic acid, and brassinosteroids. They have many possible functions. These include they produce distinct growth, developmental responses, remove gene repressors, and cause relatively rapid responses. Auxins are the master plant hormones. They function by influencing plant structure, development, and behavior. Auxin is produced in apical shoot tips and young leaves. One example of a plant auxin is indoleacetic acid, or IA. Auxin response genes. Auxin promotes expression of diverse genes. IAA repressor proteins that prevent plants from expressing auxin-repressing genes by activating proteins at gene promoters. One example of this is TRI1. It's a complex that breaks down repressors. Auxin transport. Auxin is directionally transported from one parenchyma cell to another. The uncharged auxin form is IAAH, and it enters the cells by diffusion. Auxin influx carrier is a plasma membrane protein called AUX1. It is located at apical ends of cells, and the charged version of this is IAA-, which requires a plasma membrane protein. Auxin influx carriers transport auxin out of the cells. Polar transport occurs when auxin primarily flows downward to shoots and roots. Local auxin concentrations are determined by the position in plant body. They respond by dividing, expanding, and specializing. Polar transport is controlled by auxin efflux carrier locations in the plasma membrane. Auxin effects. Auxin effects include apical basal polarity of seed embryos, inducing vascular tissue to differentiate, mediating phototropism, promoting formations of adventitious roots, and stimulating fruit development. Humans use auxin too. They use auxin to produce seedless fruit, to retard premature fruit drops in orchards, and stimulate root development in stem cuttings. More plant hormones. Cytokinins stimulate cell division. Cytokinins are primarily produced in root tips. At the root and shoot tips of a plant, cytokinins influence meristem size, stem cell activity, and vascular tissue development. The hormone functions for root and shoot growth and branching, production of flowers and seeds, and leaf senescence, or leaf aging. Gibberellins, or gibberellic acids, stimulate cell division and elongation. Gibberellins are produced in apical buds, roots, young leaves, and seed embryos. The hormone interacts with light and other hormones to foster seed germination. The hormone also enhances stem elongation and flowering. More plant hormones. Ethylene influences cell expansion. Ethylene role in cell expansion includes dicot seedling growth. It coordinates development and stress responses. A simple hydrocarbon gas is produced during seedling group, flower development, and fruit ripening. In the root tip, ethylene determines how many cells undergo division or remain inactive. Ethylene also has additional functions that include defense against osmotic stress and pathogen attack, and leaf or petal aging. Ethylene is also a gas that diffuses through the plasma membrane and cytosol to bind to receptors in the ER. The activated receptors inactivate protein kinase, CTR1, enabling transcription factors to induce transcription in the gene. Ethylene also helps seedlings avoid damage by inducing the triple response, which includes 1. Ethylene prevents the seedling stem and root elongating. 2. The hormone induces stem and root swelling radially also increasing the thickness. And 3. Seedling stem bends so embryonic leaves and delicate meristem can grow horizontally. And here's another table with all the major types of plant hormones. Several hormones help plants cope with environmental stresses. Environmental stresses include flooding, drought, high salinity, cold, heat, attack by diseased microorganisms, and animal herbivores. There are protective hormones. These include abscisic acid and brassinosteroids. Abscisic acid, or ABA, slows or stops plant metabolism in poor conditions. It may induce bud and seed dormancy, and ABA receptors are G protein coupled receptors. Brassinosteroids induce vacuole water uptake and influence enzymes that alter cell wall carbohydrates, also fostering cell expansion. Mutations that affect brassinosteroids can cause plant dwarfism. They can also cause cell drop and unroll grass leaves and stimulate xylem development. Brassinosteroids inhibit transcription factors for destruction. And now for the summary. Plant hormones interact with environmental stimuli to control plant development, growth, and behavior. Auxins play a role in plant behaviors, including phototropism. Auxins can be transported downward or upward or sideways in the plant. The position of the auxin influx and efflux carriers determines the direction of auxin transport. Other plant hormones include cytokinins, gibberellins, ethylene, abscisic acid, and brassinosteroids. Gibberellins function to release cellular brakes on gene expression. They also interact with cell proteins to influence plant growth. Ethylene plays a role in seed germination.